everybody, my name is Adriana Beltran, and I am assistant professor in pharmacology, and I direct the Human Pluripotent Stem Cell Core Facility at UNC. The lab works basically on generating cellular models for rare diseases. And today, I'm going to talk about our efforts generating cellular models to study molecular cellular functions of the citrate transporter SLC13A. And I'm going to share with you some of the two techniques we use and where we are in our efforts generating those models. So this slide summarizes what we do. We basically take samples from patients and reprogram those to make induced pluripotent stem cells. Induced pluripotent stem cells are cells that are similar to embryonic stem cells, meaning they can differentiate into different cell types, and they can self-renew. And they allow us to do things with them like editing their genome, creating stable modifications, and as I mentioned, they can differentiate. And then we can use these models for to model different diseases and use them for drug discovery as well. So what we do with the stem cells, the somatic cells that we receive, we use a set of 4 transcription factors that are known to maintain pluripotency in embryonic stem cells. And when we force the expression of these 4 transcription factors in somatic cells, these somatic cells acquire characteristics similar to embryonic stem cells. And they are called induced pluripotent stem cells. So how do these work? Let's see. This is how the process look like. We have blood from patients. Here we are collecting only the white cells, and we culture these cells for a couple of weeks. And then we deliver those 4 transcription factors using the Sendai virus. This virus is a cytoplasmic virus, so it's only going to induce the endogenous expression of, well, it's gonna express these 4 transcription factors, and then these will express the endogenous genes. So after 3 or 4 weeks, we will see these colonies appear, and then we will just propagate those colonies and characterize them. So far we received 7 patient samples from different individuals and harvesting different mutations, as we can see here. And once we reprogram these samples, I think everybody has seen this table. These are the 7 iPS lines we made. Here we have the 2 transcription factors that maintain pluripotency and 2 proteins that are expressed only in the stem cells. We karyotype those iPSCs to be sure that those cells maintain normal chromosomes during reprogramming. And we also perform a comprehensive characterization by qRT-PCR to prove that those cells are pluripotent and they can differentiate into the 3 germ layers, which means they can potentially differentiate into any cell type. Once this work was done, then we wanted to correct the mutation, 2 mutations and 2 different iPSCs. And I believe the scientific advisor from the TESS Foundation decided which mutations to correct. So we decided to do TESS 2 with this, the mutation, the 655 G2A mutation, and the 1475 T2C mutation. mutations in the same IPS line, and the TESS5 with the 425 C2T mutation. So what we decided to do to correct these mutations is to use the CRISPR-Cas9 system. And basically, that's it. We use that system because it's very, very efficient. This is the Cas9 protein and some of the components of the system, like the guide RNA, that will actually recognize the target sequence that we want to cut. The Cas9 protein cuts once the guide RNA binds to the target sequence. And that is all what CRISPR do. After that, the cell takes care of the double-stranded break by repairing this break using two different mechanisms. And in this particular case, if we provide the cell with a homologous template, the cell will repair this double-stranded break in a precise way. So that imagine having here that point mutation in the green part, and the cell will incorporate that. In addition to that, we use this system, a synthetic system, meaning we will deliver these components into the cells by electroporation. They will be present for a few hours and then disappear. So we will have the recombinant Cas9 protein, the designed guide RNA, and here we have the repair oligo to repair that point mutation. And then we will screen and select clones that are correct. And to the right side, we have the workflow of the gene editing. We design first the guide RNA tools, then we deliver into the cells. We do a test with a pool of electroporated cells to assess efficiency, whether we have in that pool cells that are corrected. And if that's the case, then we move to do single cell cloning, screening, and then we confirm corrected clones by Sanger sequencing. So for the first mutation that we corrected, we designed 4 different guide RNAs. And what is important here, and the reason why I'm showing it, is because this distant side is how many base pairs this guide RNA binds to where the mutation is. And this score means how the probability of this guide RNA targeting that site. And so we scanned the DNA close to where the mutation is, and we found these 4 guide RNAs. We designed those 4. We synthesized one, and after this, we designed a repair oligo. So here you can see where the red arrow is. That we are changing that adenine by adenine. And then we confirm, we translate this DNA sequence into a protein sequence to be sure that the amino acid that we want to change is there, and we are not introducing any mutation there. All these tools were delivered into the test number 2 cells. And here I'm showing just where the mutation is, that particular mutation, and 2 clones after the DNA, after the cells were repaired. And to the right, I'm showing that test 2 has 2 mutations, and to the right, I'm just showing the other mutation that still remains. So these 2 clones are heterozygous now for the 1475 mutation, and theoretically they should have now 1 allele expressing the protein. We haven't been that successful with the 425 mutation. For some reason, we designed 3 guide RNAs. 2 of them are able to target and cut the site, but the repair oligo is not repairing the site. So we have done a number of things. We designed a new oligo with different homology arms, and it's not repairing the site. We don't know exactly why, but it's just not happening. So now the last repair oligo that we designed is a little bit, it's like 20 base pairs larger to the left, and I believe 15 to the right. And we are doing other things to try to find out why the cell is not repairing that site as they supposed to. For the mutation 1475T, that we have the same issue, except that here, the guide RNA that we started working with is this one here, which is 10 base pairs apart from where the mutation is located. That guide RNA didn't cut at all. We played with different doses of the guide RNA, and it didn't work at all. So we designed this second one, and now this one is cutting, but the efficiency is quite low. So our screening, if the guide RNA doesn't cut efficiently, then the screening is quite large, meaning we have to screen more than 1,000 colonies, and that is problematic. So what we are trying to do again here is we are using a couple of chromatin remodeling drugs to treat the cells before we deliver these components and see if we can improve the repair efficiency. So far we have repaired one of the mutations in TESS 2. And as I said, these cells, we isolated 4 clones that are corrected, and these cells should express the SL13A protein, and we are working in the other 2 mutations, making some adjustments to improve the efficiency and see if we can isolate a couple of clones that are corrected. Also, in the meantime, every single line that we make, we do differentiate them into neuroprogenitor cells. We are developing a protocol that allows expansion of, for example, 300,000 iPSCs to several millions of neuroprogenitor cells. These will potentially be a cost savings, just as Gaia mentioned. You can actually do one differentiation experiment, freeze many vials of cells, and do all your experiments in a reproducible manner. So this is my conclusion at this point. We have made 7 different iPS lines, and we already distribute them to some investigators that are currently doing more biology with these cells than what we are doing. And this is the work in progress. We are still working with 2 of the mutations to correct those 2 mutations in TESS 2 and TESS 5. There is an error here. And we are differentiating the cells into neurons, different types, cortical neurons, motor neurons, and astrocytes. And with that, I would like to thank the TESS Research Foundation and Felix. Felix is this person here. He's been very, very patient with designing oligos and repair oligos and the guide RNAs and going back and forth sequencing and trying to understand why the gene, why we cannot repair those two mutations. I just want to mention that these two mutations are the most difficult mutations we have work on. We have done single point mutations, correction, and inducing mutations in around 25 different genes. And for some reason, these two are difficult. That's all I have.
Thank you so much. I wish we weren't difficult. I don't know what to say. I don't. We'll have to come up with a plan. Maybe everybody can weigh in on what the best next option is at the end. Maybe I'll add that to the chat.